Hello, my name is Matthew Leslie, and I have a microfluidic platform to probe hypoxic cell metabolism. So if you think about solid tumor growth, as a tumor grows larger, it will begin to move away from the vessel from which it was first formed near. As it increases its proliferation and increases its metabolic rate, these problems become worse so that you get variable hypoxic regions within a tumor mass. These regions of hypoxia are associated with advanced malignancy and metastasis, as well as chemo and radio resistance, so they're of large interest to oncologists. The motivation for this work is that tumor cell metabolism is often adopted in such a way where not only can these cells live in these harsh environments, but can thrive in them. Little is known about the molecular switches that govern these oncogenic metabolic adaptations, and so we're hoping that our work can help uncover some of that. Currently, not enough people are working in hypoxia because it's very difficult. You constantly have to keep out atmospheric gas, which makes reagent handling pretty difficult. So if you imagine all of your cells, media, and reagents have to be constantly kept from being exposed to open air. So to solve this, we came up with a microfluidic platform that the cells within the platform would be kept at constant hypoxia. So if you look here in the main cell chamber, the baby blue, we can load tumor cells in Matrigel for 3D culture. In the red, we flow media, and in the green, we flow gas. If you flow the media slow enough and the gas fast enough, you can form a diffusion here that creates a hypoxic environment within the main cell chamber. Here, we've arrayed our device to allow for higher throughput work. It's a simple PDMS microfluidic platform, oxygen plasma bonded to a glass substrate that can be imaged directly because it's made to fit onto a scope. We first validated this device in COMSOL for oxygen diffusion, and then we used the ruthenium-based chemical reporter RDTP, which has a fluorescent emission that is quenched in the presence of oxygen. So we loaded it into the main area of the device, started imaging it, and it wasn't fluorescent at all. As our oxygen concentration dropped, its fluorescence increased and increased until it hit a steady state at around 12 minutes at 0.5% oxygen by volume, proving that we have a hypoxic cell culture platform. In this platform, we culture tumor cells that have been transfected with a live cell biosensor. This is a single protein chromophore that the cell produces on its own. So a picture like this is not a stain. This is a fluorescent biosensing protein that is produced by the cell that we can target to any organelle, be it the cytosol, the mitochondria, or the endoplasmic reticulum. This biosensor provides a redox readout that we can use to probe metabolic, xenobiotic, or physical threats. So we've combined these tumor cell lines in our hypoxic microfluidic platform to probe different points of cell metabolism. Thanks for listening.
Hi, I'm Seth. I'm from UC Merced. And for my project, we were interested in modeling ensembles of superparamagnetic iron oxide nanoparticles. Now, these nanoparticles are small, around 10 nanometers. Now, if you have bulk iron has domains, magnetic domains, in which the total magnetic moment is points in a certain direction, and you can add them like vectors. And but if you have iron oxide nanoparticle around 10 nanometers, that's the size of a single domain of bulk iron. And that means they only have a single domain, which means like only a single direction of magnetic moment. Now, being able to simulate this would be very useful in that you can use it to deconvolute certain optical experiments, like the magneto-optical Kerr effect, in which you bounce vertically polarized light off a magnetized sample, and the the polarization of the light changes by a certain angle. This angle is very small, like a microradian, but we can still measure it. And this angle is proportional to the total magnetization of the sample. Unfortunately, a lot of times when you do this experiment, you have to do it with iron oxide nanoparticles in liquid crystal. Now, liquid crystal is also birefringent, which means that it affects the polarization of light itself. So what you end up with is a convoluted signal in which you get a contribution from the iron oxide nanoparticles as well as the liquid crystal. So this would be useful in that it would help you deconvolute the signal and take out the noise from the liquid crystal. Now, and here we have some preliminary data from these MOC measurements. And as you can see, as you increase the electric field, the Kerr angle increases. And until it gets to a certain point where it saturates out. Now, at this point, the sample has reached its saturation magnetization, which means it's as magnetized as it's going to get, and you're not going to get any more out of it. Where did I go? Okay. So there's another tool on NanoHub called ClusterMag, and this allows you to simulate the hysteresis loop and magnetic moment of any magnetic nanoparticle. So this particle has a 1100 EMU per gram. As you apply a field at around 30,000, the magnetic moment flips. It's like a step function. It has no response until it does respond. And then coming back down, it responds at the exact same threshold, which means that it doesn't remember any of the magnetization you applied to it before, which a lot of, like bulk iron does. Okay. Now I'm interested in observing this effect and seeing whether or not pushing clusters of iron oxide nanoparticles together changes this applied field, or changes the threshold. And the final result should be something like a magnetic analog of DDS-CAT, which spits out, the total magnetic, should give you the total magnetic field, a total magnetic moment, and the hysteresis loop.
Well, my name is Mohammad Kabirul Islam. I come from Bangladesh University of Engineering and Technology. I'm a faculty there. My project deals with compression of structured soils, idealized as collapse of subaggregates which fail under different compression pressures. Structured soils are soils which would have, say, for a unit volume of soil, 10 to 12 times or even more amount of water, but they behave like rocks. And when they reach a certain threshold pressure, they suddenly collapse and you have dramatic deformations. And like for oil platforms built in the ocean, sometimes these oil platforms would completely collapse into the sea because of the presence of this kind of sediments. Now, my project deals with understanding the collapse behavior of these kind of sediments, and I have assumed that there are like sub-aggregates within this particular soil aggregate which fail at different pressures. And it happens like a card house type of thing. One thing fails and the other things do not. So they keep the thing intact, then you have to give more pressure, then another thing fails, and gradually you have a sequence of failures and you have a behavior like this. I did a finite element analysis, frequency analysis, for which I developed my own equations, used the frequency analysis results in the finite element analysis, and doing a numerical numerical modeling of the soil behavior. I have come to see that structured subaggregates do explain very well the behavior of structured soils, collapsing soils that are available experimentally. So this is the crux of.
So my name is Kai Ming Cai from Department of Material Science in UIUC here. So I come from Professor Chen's group. So today my topic for the presentation is readout responsive cell assembled transient parametric therapeutics. So in drug delivery area, we are quite interested in how to efficiently deliver the drugs to the designated cancer tissues. Another issue we are very interested in is how to release the drugs from the drug delivery vehicles. Actually, if we can, very interesting aspect of my research is that we can sort of control the release of drugs by design of different trigger-responsive chemistry domains in the polymer structures we have. So here is basically the structure of my polymers. We have definitely designed a polymer chains which has the drug as the backbone of the polymers, and the polymer chain is sort of hydrophobic, and we try to through a certain chemistry we can modify the drug polymer itself by amphiphilic polyethylene glycol on it, which made this polymer drug itself amphiphilic. And very interestingly, the polymer itself can self-assemble into very stable nanoparticles, which only release the drugs in the presence of the certain trigger we designed. In this case, it is a cell triggers, which has a very high concentration in intracellular environment instead of the extracellular environment. This will make sure that our drugs will only release in the cancer cells instead of the blood circulations. Very interesting, we find that we can easily control the size of the nanoparticles by changing the method we use to formulate it. Actually, here we use a nanoprecipitation method. But in brief, it is just dissolve the polymers in a certain organic solvent that is DMF and add it into waters. So we found that when we change the polymer concentration in the DMF solutions, we can easily tune the size of nanoparticles from 40 nanometers to 90 nanometers. So, which means that we can control the size we can make a particle with designed size by simply tune the conditions we have. And from the TEM, we confirm the spherical morphology of nanoparticles. And further work we did is to confirm the trigger-responsive property of our systems. Very interestingly, we can see a concentration-dependent release kinetics for our systems. And in the absence of triggers, the particle is very stable without releasing any of the drugs in the system, actually. So we further do an in vitro assay to evaluate the anti-cancer effect of our systems. That is the MTT assay here. So we compare our nanoparticles with the drug we use that is conjugated to our polymer, and in clinical use, irinotecan, that is a drug or derivative of the drug we use, this SN38. We found that our particle system is 100 times more toxic than the clinical use drugs, and also has comparable toxicity to the drug we use as that is the precursor of the nanoparticles, which means that our system can efficiently release the drugs and uptaken by the cells in a very high level.
Hi, my name is Ariel Ashakor, and this is my project. So my work focuses on shape memory, mechanical actuation of polyelectrolyte multilayer wrinkling. So you're probably wondering, what is shape memory and what are polyelectrolyte multilayers? So if you can imagine that my hair, for example, is a polymer, we can heat this polymer above its glass transition temperature, deform it, so elongate it to a new position, cool it at this new position, and then it will remain at this position. But as soon as we heat it back up again, it will elastically recoil back to its original position. And so that's the basic concept for shape memory polymers. Now, polyelectrolyte multilayers are these nanometer thin, controllable films, and so that contain a polycation and a polyanion, or a positive charge and a negative charge. And so what we want to be able to do with this project is to understand some of the effects of surface substrate stiffness, chemistry, and topography on cell behavior. And so what we do is we go through the basic steps of what I just showed with my hair to make the shape memory polymers at a new length, and then we coat it with polyelectrolyte multilayers. And so we can change the charge on top of our substrate to be either positive or negative. And then upon heating the substrate back up above its glass transition temperature, Then the polyelectrolyte multilayers compress and we get a wrinkle topography as shown here. And so once that also happens, there is a optical change in the polymer substrate. And this is how we initially know that we have wrinkles. So from that we can do optical microscopy, we can do AFM, profilometry, and really try to characterize what's the amplitude or how high are the wrinkles, and what's the wavelength or how spread apart are the wrinkles. And this will all dictate how well cells can align on our substrate, which can mimic some cellular environments, like in your arteries. And so what I did is I strained the shape memory polymer to 3 different strain percents, and I varied the rotation speed to create different thickness of the polyelectrolyte multilayers, and also in a different stiffness of the polyelectrolyte multilayers. And as you can see here, as the polyelectrolyte multilayers become more thick, they have more dense wrinkle pattern, and there's less space between each wrinkle. And this changes the roughness of the surface, the contact angle, which all affects how the cells will spread onto our substrate. And so what I did was I characterized the wrinkle wavelength, which there's a linear increase with the strain that we apply. And the wrinkle amplitude is around 50 nanometers, but the wavelength is around 1 to 3 microns. And then we characterize the roughness, which also varies with the strain that we apply and the rotation speed that we apply, the contact angle, and the light transmittance. And so in the future, we hope to be able to use this substrate with endothelial cells and osteogenic stem cells in order to understand some of the dynamic effects of changing 3 variables that are very important for cell differentiation, behavior, and motility.
My name is Kyle Chisholm. I worked for UC Merced on my project here to create a inexpensive UAV for fire perimeter control and monitoring. Basically, what CAL FIRE asked of us is that they would like an inexpensive, small and portable plane that could be used to determine the perimeter of the fire. And so what we did is we went out and got a fairly cheap remote control RC, which you would see at your normal park or playground or anything like that, and used an Arduino, or for our electronics package we used an Arduino, an IMU, GPS and micro SD to actually record and control our airplane. The way our airplane works is that it has a thermopile, which is a temperature sensor. It reads a certain cone width of area on the ground, and it needed to register temperatures above 300 degrees Celsius because the flash point of wood is 300 degrees Celsius, so as soon as it hit 300 degrees Celsius, it knew that it was above the fire. And so what we did is we decided to do a zigzag-like pattern over the fire, going hot and cold spots. So our plane comes in to a fire, realizes that it's over the fire, and then will turn until it hits a cold spot, which means just below 300 degrees Celsius, and continue going hot and cold, making a zigzag-like pattern around the perimeter of the fire. And after that, we took an average of our perimeter, and we used that as our basic model for the perimeter of the fire that the fire department could use. So what we want to do next with our work is we'd like to look into other horizontal and vertical markets, such as agriculture. We could get an actual thermal camera on there if we wanted it to be a little more expensive, and actually read crop temperatures and decide if they're accurately, or if they're watered or unwatered. We also want to look into volcanoes. Flooding would be amazing, and especially emergency relief for finding distressed people in the woods, lost children, any sort of emergency that it may be. And if you'd like to actually visit us, you can go to our website, www.firesitesystems.com, and you can check out more.
I'm Anders Henriksen. I'm from the Technical University of Denmark, and at the Technical University of Denmark we have a project called Aquavia, where we are using aptamers, which I'll come back to detect viruses in running water. So we have this lab on the chip system that uses aptamers to detect the virus in the water. And aptamers are DNA sequences that bind with itself. So it's like a single-strand DNA which creates like a three-dimensional structure that will target a specific virus. So the shape, which is the important part of DNA, depends upon the base pair interaction of DNA, like the AT and DC interaction. That means it's very temperature dependent. So if you have one aptamer or one DNA sequence that works inside the human body for cell targeting of disease targeting, then it might not work in running water, as that's a different temperature and different salt concentration. So I'm doing a test system to figure out in what conditions a given aptamer will work. So we do it in this way, where we have a magnetic sensor and magnetic labels, and then in between we bind, we have the virus, which will act as like it's in the semi-assay between the sensor and the labels.
When you come to this point,
why we do this and not, usually you would do a fluorescent label instead of a magnetic label because that's easier. But we want to do real-time sensing, and that means that we cannot use fluorescent labels because they photo beats over time and it's generally unstable. So the magnetic labels, they create magnetic fields that can be picked up by the magnetic sensor, and that magnetic field corresponds to the presence of viruses. And then we have our experimental setup, where we have this magnetic sensor, which you can see over here. It's targeted in between here, and then we have this top, which defines the microfluidic system and the electrical contact to the sensor. We also have a temperature controller and a PVC tube where we can attach syringe and syringe pumps. And that way we can sweep the temperature and we can sweep the salt concentration while we are monitoring the interaction between the magnetic labels, the virus, and picking up by the magnetic sensors in real time. And then we can like trace out, say we have one system or we have one condition where it's working, and then we increase the temperature and we can see when does it fail, or we can increase the salt concentration and see when does it fail. And then we can trace out like a phase space for in what condition is it working. We haven't made it to this point yet, but we have done some preliminary tests where we've done DNA melting curves, where we have magnetic labels covered with DNA, and then we have immobilized complementary DNA on the sensor surface. And then by having sensors with the complementary DNA and a mutant DNA where one base pair exchanged, we can see that these two interactions fails at different temperature, which is generally known as like a melting curve when we sweep the temperature. And we hope to convert this approach to also work with aptamers and this virus interaction with aptamers. And when we have done that, then we will know the aptamers which has been discovered, when will they work, and can we use them in this project for detecting virus in drinking water.
Hi, my name is Heather Chimori, and my poster is on the effects of radiation exposure on gallium nitride semiconductor metal ultraviolet photodetectors. And essentially the UV photodetector that we're working on is a semiconductor metal architecture, and it's used for sun sensors. And basically it's a binary response for satellites. Am I looking at the sun or am I not looking at the sun? And in tandem with a bunch of other systems, it can help orient the satellite to the proper
orientation?
I'm working with a material called gallium nitride, and it's a wide bandgap semiconductor material. It has a bandgap of 3.4 eV. When you alloy gallium nitride with aluminum gallium nitride, at the interface there's a two-dimensional electron gas layer that's formed. So this sea of electrons could be used potentially to enhance sensitivity of sensors. The other advantage is that gallium nitride can be fabricated on silicon, and so we can fabricate many different devices on the same platform using established microfabrication tools. What I've done with the photodetectors is place them in a radiation chamber and looked at how the behavior has changed as we increase the dose to 50 kilorad. And the initial results are very promising. The current appears to remain stable even up to 50 kilorad, and so this seems like a promising material and promising sensor for UV for this particular application. So, for future work, we're looking at incorporating different nanostructures into the device as well. So.